All right, well, it's 11 o'clock. Hopefully, I know there wasn't much promotion for this webinar, but we will do our best to get promotional things out after the webinar. We are recording. And so this will be shared on hopefully on our North Central Region Water Network website. And like I said, we'll put some more promotional things out there. So this will get viewed, I'm certain, by many different industry and academics and things like that. So we can get started. My name is Amy Weckel, and I'm from the Illinois Water Resources Center at the University of Illinois at Urbana-Champaign, and I'll be your moderator today. The webinar is hosted. Let's see if we can forward this slide. Sorry. Webinar is hosted by the Algal Bloom Action Team, of which the Illinois Water Resources Center is a member. The Algal Bloom Action Team is a collaboration of water professionals, researchers, and educators from 12 states in the North Central region of the United States. Team members include the National Network of Water Resources Research Institutes, or WRRIs, the North Central Region Water Network, and University Extension within each state in the North Central region. In addition to this webinar series, our team hosts a website of resources, including fact sheets and frequently asked questions on HABS topics. I encourage each of you to visit our website, which I'm going to put in the chat right now. Ta-da. And there you can explore some of the resources we have to date, including the fact sheets that I mentioned on what you should know about HABs and their health-related effects on animals, prevention and treatment for landowners, and a HABs frequently asked questions database. There's also a link to the recordings from previous webinars, as well as our virtual HABs research symposiums, which are held annually in January. Before we get started, just a few housekeeping items. We will have a Q&A session after our speaker presents today. Please list your questions for our speaker in the Q&A panel, not in the chat box, and we will do our best to address all of the questions following the presentation. If there's a question already posted to the Q&A and you have the same question, please upvote it, and we'll be sure to get answers to the most popular questions during the session. Today's presentation is being recorded, as I mentioned, and will be posted on the Algal Bloom Action Team website following today's session. If you're having any technical issues or have any questions about the Algal Bloom Action Team, please list those in the chat as well, and we'll be happy to assist you as best we can. Our speaker today is Cody Sheik. Cody is a microbial ecologist who focuses on microbial life in freshwater systems. His lab uses molecular tools to address a broad range of questions regarding nutrient cycling and cyanobacterial blooms, cyanotoxin production, biogeochemistry, and biogeography. I will stop sharing my slides now, and Cody, you can take it away.
All right. Howdy, everybody. Let's see. Everybody can hear me just fine?
Yes, we can.
Awesome. Well, thank you all for coming and participating today. And I'm excited to share some of the work that our graduate students have been doing across Minnesota and in a couple other spots as well. I'm an associate professor at the University of Minnesota Duluth. My home department's in the biology department here, but I'm also a member of the Large Lakes Observatory. And one of our, as our name implies here, we all study large lakes of Earth. And so you can see on the right-hand side over here, we spend a lot of time across the Great Lakes thinking about current phytoplankton communities and other microbial assemblages, but also thinking about phytoplankton from the past. And so we do a lot of sediment work and are doing a little paleo reconstructions as well of phytocommunities. And I'll touch a little bit about Superior today, but not a whole lot. What we're going to focus on is mainly Minnesota, because I think it has a lot of broad applications to what everybody else is studying in blooms and whatnot. But before we get started, I really want to do our acknowledgements up front here. And a lot of this work is coming from these two fantastic former graduate students. We have over here in this picture is Leah Schleppenbach. She was a master's student. She graduated a couple of years ago, and now we kept her around Duluth, and she's still working and is a fantastic tech. And then Kayla Netwara, who took a job here for managing waters in northern Minnesota, but is finally going to be defending her PhD next week. So I'm very excited to see both of them doing really, really well. I also want to acknowledge my co-PI for a lot of this work is Chris Philstrup. He's at the NRI, which is the Natural Resources Research Institute here in Duluth. And we also have a whole slew of other people that helped out with analyses and whatnot. And the majority of the funding for this work today is going to come from Sea Grant. And they've really helped push several of our projects now and are really a fantastic resource for us. So with that said, let's start here. I always kind of start off with this slide, whether it's just talking about bacteria or just cyanobacteria, just in general. It's because I want to bring it forward to everybody that despite the size of these organisms, which are microscopic, they can be very powerful on the entire stage of, say, of Earth. And so these cyanobacteria have been around in one form or another for over close to 3 billion years. And at some point, they evolved the ability to take energy from light and put that through your photosystem, fix carbon dioxide, and then also make oxygen. And because of these organisms and their blooms 2 billion years ago in the ocean, we now have the ability to take deep breaths and whenever the smoke's not happening up here. But these organisms have evolved to be really indisposable for most aquatic systems. And that's because they take these inorganic forms of carbon, so like carbon dioxide or say dinitrogen gas from the atmosphere, convert these things over into bioavailable forms. So like dissolved organic carbon, bioavailable nitrogen in terms of like, say, amino acids or ammonia through nitrogen fixation. But one of the reasons why we're all here is also because of these secondary metabolites that they like to produce. And this is really one of the driving forces of why we're studying cyanobacteria and why cyanobacteria in general are probably one of the most studied organisms on the planet, yet so poorly understood still. And this is one of our lakes. I won't talk about this one today. This is Lake of the Woods. This is on the Canadian border. So we have Canada up here and then Minnesota down here. But you can see the extent of these blooms that can happen. And this is an Aphanizomenon bloom from 2021. This bloom occurs almost on your watch. It's probably going to be starting up here soon. Now it's starting to turn over into late August. But this thing blooms and it takes up the entire basin. And this is one of Earth's large lakes if you took it by the amount of shoreline it has. And so these organisms can really enact big change on very broad scales. And so across the board, we're seeing blooms, I say board, across the Earth, we're seeing these blooms increase globally. And some of this may be due to our ability to recognize blooms, right? So as water quality has gotten better, our ability to see blooms and recognize them and study them have also increased. But we also know that for lakes like Erie, these blooms are actually real. It's not just observations. We can see these things growing across the Lake of Erie. And what's interesting about this is that when we think about blooms and what's driving them, it's not really one thing that we can really pinpoint, right? We've spent a lot of time thinking about phosphorus. We spend a lot of time trying to control phosphorus, right? Control nitrogen even now. But yet we still see blooms happening. And so there's this combinatorial factors that have to come together for these blooms to proliferate. And it's different. So here, if we look at like Lake Erie, we see this like an improvement in bloom intensity, but then also this big increase as microcystis is really taken over. Like Huron has seen this steady decline, while Lake Michigan sees this really stochastic, really no significant trend, although we do see blooms happening in Michigan as well. And so What this says is that there's a whole slew of factors that could be coming together to provide the right conditions for these microorganisms to proliferate as blooms. But we do know that from some great past work is that temperature really plays an important part. And so globally, we're seeing in these temperate lakes warming. And even in big lakes like Lake Superior, which I think is somewhere over here in this really high side, is that as we're increasing our global temperatures in the air, we're also seeing the waters increase as well. And so these lakes like Lake Superior are changing thermally, which has huge impacts on the organisms that are living there. And here in Minnesota, we're seeing changes in temperature as well. So over here in this little cartoon, you can see that these are winter low temperatures. So these have been recorded since 1895. You can see that we've had around a 5-degree increase in winter low temperatures in the southern part of Minnesota. But while we're up here at the very top, just for reference, Duluth is right around here. Up here, we're seeing almost an 8-degree Fahrenheit change in winter low temperatures, right? And, this doesn't really, if you're thinking that it's minus 40 in 1890, and now it's like minus, I don't know, 32, those temperature differences don't really matter as much, right? It's still bitterly cold. But when we start thinking about the shoulder parts of these seasons, that's where this really comes into play. And so because we're seeing this warming, we're seeing a decrease in the amount of ice days that we have here in Minnesota. And so in the north, we have at least 10 days of ice cover that we've lost. And in the central lakes, we've lost over 14 days. And so you can see this nice trend. It's not nice, this big trend here. And this is really being driven by these annual temperature increases. But from thinking about this from a productivity standpoint, what we're seeing here is not just that we're losing ice, but we're also losing, we're gaining time for organisms to grow, right? So our growing season is potentially lengthening, right? And so that gives us more time to interact with these organisms, more time for them to grow, more time for nutrients, yada, 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 right? And so this is where we're sort of sitting in Minnesota, is that with these decreases in ice, we have this now more chance to be interacting with these organisms. On the other side of that, because of climate change, we see that rainfall is increasing across the state, especially in our southern portion of the state here. And what's interesting is that these sorts of patterns were predicted 20 plus years ago, but we're seeing that the intensity and frequency of these rainfall events are increasing, right? And so what this means is that we're seeing these big nutrient pulses delivered through sediments and just runoff being pulsed into these water systems more frequently, right? And down here, just as a visual, this is Lake Superior after a really heavy 100-year flood. And you can see the amount of sediment that is coming from our central river system here, the St. Louis River, and being pushed out into the lake. And so you can think about, not only are we delivering sediment materials, so sand, silt, clay, along with those clays and those colloidalness, since clay is charged, you're also getting this big pulse of nutrients that are being absorbed to it as well and slowly released out into the lake. And so in lakes like Superior, where it's very oligotrophic, you could be having this huge pulse of new nutrients coming into the lake that could help stimulate food webs and phytoplankton. All right, so when we look across our lake systems here in Minnesota, we see that we have this huge transect of different types of habitats. And I don't know if anybody's driven across Minnesota. I've had the privilege this summer to drive across it. It's big, it's vast. It takes a long time to get from down here in the southern part up here to the northern part of the state. And because of that, we have a very heterogeneous landscape. And we have agriculture dominated southern and western portions. As we transect sort of north and east, we move into these wetlands and these forested areas. But some of our lakes are in these really heavily populated areas, like this one called Peltier Lake here. This is on the northern side of Minneapolis, St. Paul. And so we have this huge gradient of lakes. And what's interesting is that we're seeing blooms happening in weird parts. So this would be Up here in the Boundary Waters area, this lake, I think it takes 2 portages to get to. So it's not an easy lake to get to, but it's experiencing blooms. It doesn't really have a whole lot of people around it. And this is generated from Adam Heathcote and his group. Oops, sorry. We also have lakes down here by Minneapolis, where Peltier Lake, where we have microcystis and dolichospermum blooms. This is a heavily urbanized lake. You know, you would almost expect it to bloom, I guess, but like we're still seeing these big blooms happening here in these urban centers. As we move up to Lake of the Woods, we have these big Phanazomenon blooms. What's interesting about this is that it's a low, it's always a nitrogen limited system. And then on the flip side of this, we have Lake Superior, which is extremely oligotrophic, has a real high offset of nitrogen to phosphorus. And we're seeing these blooms of dolichospermum here, right? And so Minnesota provides this really great gradient of different lakes and systems really to study cyanobacteria. And so this is what we're trying to leverage here, is trying to understand all these different systems. But I really want to point this out, though, is that I kind of pointed out that some of these blooms are driven by dolichospermum, some are driven by a Phanazomenon. Typically, when we look under the microscope, and many of you may have done this, is that when you look at these populations that are living in these blooms, a lot of times it's not just a single organism, like, say, microcystis. It's typically a polycyanobacterial group, right? And so this was a small bloom in the Duluth Harbor a few years ago. This one was important because it was the first time we measured microcystin. Here recently, we've just confirmed that it's microcystis that's producing some of this, the majority of this microcystin. But when we looked under the microscope of this bloom, this is what we saw, right? So you see these big packs of microcystis. We see all these different types of dolichospermum. Some of them are very twisty, some of them are really bunched up. We see other types of groups that are really long chains as well. And so when I look at this, and I'm really coming into cyanobacterial blooms really late compared to many people. When I see this, I really want to know exactly which of these organisms, which species is driving these, like, say, cyanotoxin production. While lake managers may see, or water managers may see this as saying, okay, we have the potential of all these species to produce cyanotoxin. But what we know is that some species can't produce this, and it's all really related to their genes, right? And so for a cyanobacteria to produce these cyanotoxins, they must have the genes in their genome and the ability to express these genes. And so what my lab typically does is we look at the genomes or DNA of these microorganisms, right? And so we can really hone in and identify, say, anatoxin-specific genes or, say, microcystin-specific genes on these genomes, and then relate that over to who is potentially synthesizing these really complex compounds. And in Minnesota, our primary one is microcystin. However, we have some lakes that are being impacted by anatoxin and saxitoxins. And then cylindrospermopsin is an emerging toxin, as we have cylindro being migrating north as these temperatures have also increased. So we have this really complex group of cyanotoxins to really worry about as we're going through here. Okay, so it feels very simple, like short amount of time, but now I'm looking at it like it's just it feels like a few years ago. But we started this study in 2020 to really start thinking about these distributions of lakes and the organisms that are living in there, but also to think about linkages. So if we have lakes down here in the southern portion, do they share any sort of similarity with lakes happening up here in the north? And so to do this, we took a holistic sampling approach, which I think a lot of people do. This isn't unique to just our study, but we wanted to study all the nutrients possible, knowing that nitrates and phosphorus are really big drivers of blooms. We also wanted to look at microcystin concentrations, and we're doing this in 2 different ways, one with ELISA kits and the other with mass spec, and thinking about, are there differences in congeners across here? We also wanted to look at some of the functionality of some of these groups of cyanobacteria. So we're looking at nitrogen fixation. We also do taxonomy via microscopy, and then also taxonomy with DNA. So here we use things like 16S rRNA gene sequencing. So this is just basically looking at one single gene that we can really hone in on the taxonomy of organisms and look very broadly about where these organisms are distributed across the landscape. And then finally, we look at microbial functions using RNA sequencing. So for the central dogma is you start off with, like, say, a genome, and then you have transcription of that genome, and you finally get to proteins, right? And so we're trying to look at this, what genes are being expressed across this gradient of lakes. And we did this both biweekly sampling and or monthly sampling for 2 straight years. So we have this really nice spatial gradient. We also have this nice temporal gradient, both like within the year and then also across years. And so some of the sort of the broader data here from some of this is that as we're going through here, we generated, Kayla and Leah really generated a lot of data. And so we've been trying to, like, hone it down and try to figure out what needs to go in publications and what's not. And so I'm going to give you kind of like a broad overview of some of the highlights here. But as we move from southern lakes over into more our central and northern lakes, we had these really big trend where our 2 southern lakes here in blue, this is Carey Lake, and then we had Peltier Lake here in yellow, really stood out from the rest of our lakes. So all the other lakes kind of fall in here. And this is just total phosphorus concentrations here on the x-axis, and then y-axis is just total nitrogen. And so we've drawn in different stoichiometric gradients here. So we have a Redfield ratio. We have the 20 to 1 N to P from Guilford and Hecky, and then also the 50 to 1, right? And so anything above 50 to 1 is considered to be phosphorus limited. Anything between, say, 20 and 50 is this balanced growth, and anything below 20 is thought to be nitrogen limited. And so depending on the time of year, Peltier can go from balanced growth over into limited growth. And so Peltier Lake, as you may remember, I kind of pointed it out a little bit, is this really urbanized lake just right on the cusp of Minneapolis and St. Paul. And Carey Lake is this really high nitrate concentration lake, and it's really impacted by agriculture. It's out there. And so we have these really sort of 2 endpoint members of different types of lakes. But the interesting thing is, like, no matter where we're at in terms of our northern and our central lakes, they all kind of fell on this sort of balanced growth to phosphorus limited style growth, which was really, really interesting. And so as we started looking at the microbial assemblages, or the cyanobacterial assemblages across here, we found that there was lots of spatial and temporal variation. And so we saw year-to-year variation. So here in the graph, we have years of 2020 and then 2021. And each one of these graphs is just the percent composition of that community. And you can see all these different colors represent all these different cyanobacterial species here. This upper panel is just biovolume. So you can see that we have Peltier Lake is by far our most productive lake in both years, followed by Carey. And you can kind of see the other ones sort of fall into date. We see within here seasonal variation, and so you can see how these populations are quite dynamic over time for some of these lakes, and where you have big turnover events occurring. So, like down here, I'll point out this one where we have dolichospermum groups being almost completely replaced by microcystis later in the year. And so there's so much data within here that we've been trying to really hone in on what really to point out. But it's always good to have more data, I think, and which is great. And so let's break this down just a little bit here and just focus in on those 2 southern lakes. And so because they were quite different between P limited and the N limited. And so when we look at Carey Lake, which is P limited, we find that this is a very diatom dominated lake. And you can see here in blue, these are all diatoms. And then at some points during the year, we have cyanobacteria that tend to bloom a little bit later on here. But for the most part, it is very much a eukaryotic driven assemblage. However, if we look over at Peltier Lake, which is inlimited for parts of the year, we see that it's mainly cyanobacteria that are really kind of taken over here. And two of the big culprits are microcystis and then Dolichospermum species. And so if we break this down even a little bit further here, we can start thinking more about who's actually there. And so Carey Lake, for that little tiny bit of cyanos that we do see, are primarily Pseudoanabana, limnetica. But then, as I pointed out earlier, we had that little tiny blip of cyanos, and that was driven by microcystis species here. So primarily originosa. And here, and we've changed over from percentage to just biovolumes over here. Sorry, I should point that out. And then over in Peltier Lake, you can see that early on we had Dolichospermum being very dominant. And then we had this almost role reversal where we had this microcystis take on and really bloom for the rest of the year, kind of be present there. And we had a little bit of a phenomenon towards the end part. And you can really see the differences between these lakes just by looking at them. So Peltier is very green and kind of gross. And Carey over here is still pretty clear overall. And so one of the things that we think is happening here, where we see this Dolichospermum being dominant and microcystis taking over, is that this lake can be prone to turnover. And so it's a high productive lake. And so we think that some what happens is that we have bottom water anoxia that helps release soluble reactive phosphorus, which could be driving some of the microcystis as it starts to take off. But what's also fun is that we've been measuring nitrogen fixation rates within all of these too, is that Dolichospermum, we measure nitrogen fixation throughout the year, but as Dolichospermum sort of takes a dive in terms of the population, we see nitrogen fixation rates really kick on. And so we think there may be some sort of interplay here where Dolichospermum, despite not being a real dominant member of the community anymore, is still able to functionally really do a lot of nitrogen fixation and may be supplying nitrogen for parts of these other organisms, like say the microcystis. And because we have these two really nice methods of both like the microscopy and also the DNA-based tools, we were actually trying to like go and compare these two results together. And so we're trying to do this integration of microscopy and molecular analysis. And what we see with this in both methods is that we see down here is the 16S gene. We see Dolichospermum kind of kicking on, and then also all these different types of other cyanos kicking on as well, but we also see that microcystis blooming here as well. And so I think we still have a lot of way to go here, and we're seeing a few pitfalls. There's a lot of stuff with visual ID that is really dependent on who's performing the counts. They need to be very trained to identify some of these different species. And if we look at, like, say, 16S taxonomy, is really dependent on the database and the region of 16S that you're using. Here we're using the V4 if you're really interested. But the really downfall, I think, of converging both of these types of methods is that the microscopy and 16S databases aren't taxonomically aligned. And so what we find across the board is some organisms being called one thing by visual representation, and then being completely called something else in our 16S databases. And so it's going to take a lot of work, I think, to like help resolve some of this, but it's something that we're actively really starting to think about more broadly across the cyanos. And so pulling back out, so those were just two of the lakes. And here I'm going to show a little bit more of the 16S data, is that, when we start looking across all these lakes, we can see there's some really dominant organisms in different lakes. So we see like cyanobiums, and here in this light green towards the bottom are quite predominant in most of the lakes, with the exception of Peltier here. We see things like microcystis kicking on at different times across the board. and it always seems to be present at least some sort of abundance in most of these lakes. We still see our Aphanosomnon and our Dolichospermum also being present here as well. And this is just the top 20 species in our lakes. But you can see that in some cases, they're reaching quite high abundance relative to in that entire population. So almost 40%, almost 50% is cyanobacteria in this one sample that we took. But I think the really interesting thing here about this is not just like saying, oh yeah, we see all these different groups, is that we can really hone in and really think about linkages between these lakes. And so we can really go and move beyond to say, oh, this species is present by looking at subspecies. And what we do is we call these ASVs, or amplicon sequence variants. But what this means is that you can have, we can really get down to like single nucleotide changes. So like an A switched over to a G may be able to determine two different populations of, say, microcystis between lakes. And what we're finding here, despite having this huge gradient of lake conditions and locations, we're still finding some of these species are linked. So we're finding the same organism potentially across all these different lakes. And so there's about 47 of these ASVs that are actually shared amongst all 6 of our lakes, which I find very just amazing, right? And here we have just like, we also find that there's also unique ASVs amongst all these. So like Peltier Lake has its 33 unique ones. Cary has 28 unique ones that are not found in any other lake. And so we are seeing this variation amongst these different species being present across the lakes or being shared amongst these. And so we're still trying to figure out some of these patterns here. But the interesting thing, and one of the interesting outcomes of this, is when we started looking at the microcystin congeners, right? And so being able to go in and not just say the total amount of microcystin present in these lakes, but also coming in and being able to show that there are differences amongst these congeners has been sort of a game changer for us in thinking about the physiology of these organisms and potentially what subtle variation in their genomes might be. And so here you can see that our northern lakes up here on top are really dominated by the LR version of microcystin, while we switch over here to more of an LA, LR for these middle lakes. And then as we get down into our southern lakes, the LA and then also the LR is quite predominant. What's interesting is that each lake is producing microcystin at different concentrations. So you can see up here in our northern lakes, it is a very low concentration of microcystin, but it's there. And down in our southern lakes, we have much higher concentrations, especially in Lake Peltier. Again, this is a very eutrophic lake, and so it makes sense that it's producing a lot. But when we start thinking about more broadly about the presence of these microcystins being in some cases in these really low nutrient systems where we're not seeing a huge amount of algal biomass, makes us start to wonder more broadly about water quality and being subjected potentially to low concentrations of microcystins for long periods of time. We know these things have can affect the liver and several other organs. And so you can imagine if you're constantly being exposed to these, either through eating fish or drinking water and things like that, there may be some long-term health problems that could persist over time, right? And so this is one thing that we're trying to think about more broadly, is why we're measuring concentrations in these lakes, and really getting into like the weeds of like who's producing them, right? And so one of the things that my lab's been working on is really trying to think more broadly about differences in genomes. So if you have two of the same species, what are the differences between these two organisms? Are there differences genetically between these two different organisms? And so one thing that could be help driving these different types of congeners is this idea of microbial strains. So if we think about a species, so like say Microcystis aeruginosa, A lot of times you'll have like a strain designation at the very end of it, PCC, blah, 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 blah. And that's telling you where it came from, potentially, who isolated it, things of this nature. As microbiologists, we understand that those strains, while they'll share a lot of the characteristics of the genome, there could be subtle variations amongst the genomes that could help change expression patterns of things, or even like just knock out genes altogether. And so to help better visualize this is a quick study that we did here in Lake Superior. So if we look across the board at, say, all the Great Lakes, we can typically find Dolichospermum hanging out in the water, especially Dolichospermum livermanii. And so you can imagine we have this species of weird-looking AI-generated pickles, but we have this Dolichospermum livermanii. But we know that if we go to each lake, we can pull a different strain of it. And what this means is that each one of these lakes, we know, differs by temperature, differs by nutrients. So we have, like, Erie being really eutrophic, while Lake Superior being very oligotrophic, right? And so we know, based on just, say, thermal structure, just in general, it's just a fake graph I made. This would be temperature and maybe, say, growth rate, is that each one of these strains may be adapted to different temperatures or different nutrient conditions. And so you can imagine that, like, being cold, and from Lake Superior, we have a different temperature response than, say, an organism growing from Lake Erie, which gets way warmer, right? And so when we start looking at the genomes, what this really looks like is this, where we have here, like in this green box, we have T, and down here we have an A. And over here we have A's, and here we have a G reversal. And so when we look across the entire genome, we can really start thinking about these different patterns of these different strains, and what happens when you have this reversal of a T to an A or a G to an A, right? This can do a lot of stuff with translation and change up the differences of the gene potentially, or even stop the gene at weird spots. And so we can use multivariate statistics to really help us visualize these patterns. And so as a proof of concept, we did this across the time study in Lake Superior. And what we were able to find is that we were able to pull out different strains from our little time course that we did. So we have strain 1, strain 2, strain 4 down here, and this minor strain 3. And what's fun is that this was really set up by time. So early in the season, on the 20th of July, we had primarily this strain 1 coming through our sampling transect. And then we had this big turnover from August 10th, primarily being this strain 2. And then finally in August, we had a mixture of strain 2, strain 3, and strain 4. And we didn't have that strain 1 being present at all in our samples. And so, thinking about blooms and things like that, what are the big, the implications of this, right? And when we started looking at, like, say, these different genes that were being interrupted from some of these changes, we're finding that each where these single nucleotide polymorphisms are showing up can be quite different. So here in strain 1, we found things like in the plasma membrane. We also found things in, like, nitrogen fixation. Nitrogen fixation is a big one for a lot of these. And so you can think about these different functions being affected. And so if we think about cyanotoxins being affected, it may be that they're, preventing maybe a specific amino acid from being synthesized that's then can't get incorporated into, say, microsystem. And so potentially we could be seeing changes of these congeners, not just based on presence absence of microsystem genes, but also on the regulation of things like amino acids or just the genes themselves for it. And so I think one of the implications of this is we started thinking broadly about blooms and trying to predict blooms, no matter what the system is that when we lump blooms into these generalized species categories, like, say, if this is a Microcystis aeruginosa bloom, we're missing a lot of these subtle nuances that could be helping drive the growth of these blooms. And so when we try to make these broad predictors, we may be failing because we're not looking at a high enough resolution of these organisms, right? And so part of what we're moving on for or thinking about more in general is really how do we incorporate this idea of strains and um, their potential into more of like a bloom predicted network. And so as we start thinking about these drivers and or these set of environmental conditions that make it favorable for blooms to happen, we're starting to think more broadly and change up our ideas. So instead of, I say change up our ideas, trying to like think more broadly about the organism and not just like thinking about the suite of conditions, right? So and a different way of saying that would be like you could have the perfect environmental conditions, but it's really still about the organism there and can it respond to those conditions and proliferate, right? And so one thing that we're thinking of is that you could have all the right conditions, but you don't get a bloom, which happens quite often here in Lake Superior. And so these interactions with the environment may also be driven by interactions at the total community level. And sort of help to dictate this is like, here we have like say our cyanobacteria, and they're trying to get at these nutrients, right? Whether it's carbon, nitrogen, or phosphorus. But there's this whole other groups of organisms, not just bacteria, not just other cyanobacteria, but also phytoplankton and things like that, that are also trying to go after this finite amount of nutrients within the system, right? And so it may be just like thinking about, oh, we have plenty of nitrogen, we have plenty of phosphorus, the temperatures are just right. It could also be that we have all of these antagonistic sort of effects happening within this community, right? And those sorts of dynamics are really what's helping drive blooms in some of these lakes. And so myself and several other people being led by Andy Bramberger from Environmental Canada are starting to think more broadly about these lakes and thinking about why do we see some lakes that seem to be resistant of cyanobacterial HABs? Oops, not sure what just happened there. I think it's maybe on a timer or something. We may find some that are resistant. We may find things that are more susceptible, and we find some that are prone. And but at the very heart of this is that there may be only a few that we can actually predictably predict and may be able to accurately predict based on what we know, right? And so this is all in prep right now. We're working on a manuscript that kind of outlining some of these ideas. But you can kind of think of that. Some of these are going to be really hard to predict, like say Lake Superior, where we don't have this rapid occurrence, but we do still see blooms happening. And so why is that? And is there some sort of other framework outside of just say nutrients that we can start to think more broadly of? So with that, I think I'm about out of time here. And so I'd like to take any questions you might have. I was at a wedding on Lake Superior, and all I had was my champagne cup on the beach, and we just happen to have a small dolichospermum bloom here. So you can kind of see what it looks like under the naked eye, I guess, in some spots here. So thank you all so very much for listening to me ramble, and thank you again.
Thanks, Cody. That's great. That's really interesting. We don't have any questions in the Q&A, but I kind of have one. I was wondering if you've looked into, so you've done all this genotyping and everything. Have you looked into like the ancestral sequence of any of these? Do these types of bacteria, do they produce toxins naturally? Is that the ancestral state, or is this something that's developed?
I think others have done that where they go back in and do the gene reconstructions over time to say like when would like a microcystin gene have evolved, right? And they find that it's very long lived in these genomes, dating back, predating most times, like say eukaryogenesis and just basic eukaryotes, right? So this idea that these things are new gene systems or things like that, and are here just to harm us or other eukaryotes is just probably doesn't hold, and they were probably evolved for some function, right? And I think right now that's the big looming question in terms of like these genomes and these genes and are basically why they have these, and I think if anybody could figure that out, we'd have like a real golden key to understanding cyanotoxin production and things like that. But yeah, it's still just like a big looming question, I think, for most of us.
Would you say a lot of the changes genomically are affecting the portions of the gene or the amino acids that bind the nutrients at all? Have you noticed that at all?
So in some of the ones for the dolichospermum one, they were changing some of these, I think ATP driven transporters, right? So whether or not it's actually making them more sensitive or changing up the kinetics of those is a big question mark as well. So to answer those sorts of questions, you would really need to be having these cultures growing. And for that D. limmermanii, it just hates coming back into the lab. Like we've been trying for 6 years now, it seems like. And we'll get it to grow for a while and you can see it like just like these little colonies, and then
Mhm.
one day you'll come back in and they're just dead, like they're just like
Mhm.
done. They're like we've had it. And so we
Mhm.
haven't been able to find that like magic recipe to keep at least these ones growing in the lab, unfortunately.
Interesting.
But that's like one of those fun questions I would love to ask. Like, does
Mhm.
this change in composition also change, like, say, the km of like, or the affinity for this thing of bringing it into the cell and things like that?
Mhm. Mhm. Yeah, and is it binding synthetic fertilizer or nutrients from synthetic fertilizers differently than
Right.
you know, natural? Yeah.
Mm-hmm.
Anyways, all right. Well, thank you again for your time. I'm gonna well, I do have Cody's email I can share if I can share my screen again real quick just to get this on the recording. And so I'm sure it's okay if you reach out to Cody afterwards.
For sure. Yeah. If anybody has any questions, definitely.
Okay. And yeah, so just be sure to check out the Algal Bloom Action Team website for more information. And again, thank you, Cody, for your time. And your talk was very interesting. And we'll put out, like I said, a promotional thing in case you missed it. So this gets a little more hits and things like that. But other than that,
Thank
again,
you
thank
all.
you.
Yeah, appreciate it.
Have a good afternoon.
Yeah, you too.